Go live. Okay, according to this, we are live. And we'll just see. Is that audio coming up? Okay, friends, we are live on YouTube. I know that I'm 10 minutes early, but I'm just trying to see if this is all going to work. Please type comments here. If you are joining us from the internet, you will be able to type comments online. I think your delay is looking like you have a 30-second delay or something, which is okay. That's pretty good to go back out to internet world and back to my cell phone. So. Okay, so the chat is live. We have at least one other viewer other than me playing around with it. So now the real trick is going to be at the end when we get done, if it will save. Normally, if I end a live stream, it should save it, and then that link should be active for the next user. So.
Keep my fingers crossed for you.
Thanks, man.
All right.
I'll let you know if anything goes crazy, but
Right.
I think we're okay.
Hope it's all good.
All right, friends. As far as I can tell, the internet live streaming is working. So that means you'll be able to ask me questions. People online should be able to ask questions. And then you'll be able to see this later. You think it's working? As far as it says people are watching. We're getting playback. I should be. I'm zoomed in on me pretty good. So if I write up here, so if I come over here, this is pretty cool. This is exactly what I wanted. I'm sorry. This is the only way I can get clear audio, but the audio on there should be really good. Like it should be crisp. So Steve Jobs, yeah. I will not dance too much. I don't think the singing will be terrible, and I'll get horrible reviews. But well, that usually that's for the classroom, but you know. Yeah, usually There's a lot of, yeah, I teach the majors micro in this room, and there's a lot of run and tumble. So it's big. Yeah. That's what our seminar every Friday. We have 150 people in here, so it gets pretty full. Yeah. We're spoiled rotten. So this actually is the original old auditorium used to be called Gunderson Auditorium. And it used to be stadium seating all the way to the front. And so this floor was about 3 or so feet further down because there was a stage up here. And so you actually had a stage with ramps coming off of it. And the seats were about 30% smaller than what they are now. And
Was there like not a classroom downstairs?
oh, there was.
But it was just really short.
No, it was the same, exactly the same as it is now. There is in between floors on this. Yeah, um, I think it's 14 feet between floors. So, um, so yeah, if you were to pop open these tiles in like in the hallways and stuff, there's 10 to 14 feet of space above them. It's all the space that you need to run electrical, plumbing, fire suppression. Yeah, yeah. Okay, so we got about 4 more minutes before we'll officially get started. Again, we have a few viewers online. Please type comments in the comment area. On the live stream, you can see those, right? When you pull it up online, there's a comment area. Just go ahead and write in, and we'll be ready to chat.
You've been up really like a long time, so something to do. Well, like it's probably how I did it. It's like the hour is pretty short. So he's like, I just kind of left it. What is it? My friend is
And who wouldn't and why?
Yeah. No, like what actually is a complex? Yeah. Like the actual.
Where would you go?
Welcome to the party. All right, so we will have about an hour. The next class starts in here at 4, so I want to kind of give them a few minutes to get settled. And so let's go ahead and get this party started. Welcome to the review session for exam number 2. Again, we're streaming live on YouTube, so if you have any questions, you can type those questions in the box and I'll try to get to them. For anybody in house, you can just please ask me the questions directly, and nobody can see you, so I have the camera zoomed in far enough that you don't have to worry about being streamed or anything. So, and I don't really know how well they'll actually even be able to hear you, so I might have to repeat some questions. So, let's get the party started. What do you got for me? Yes.
Number one, what is the? Yeah, I don't remember going over that. The
Tinea pisiformis.
Yes.
Yeah. Anybody remember what that was? That's your tapeworm. Yes. So the important things that we talked about with the tapeworm, again, this was wrapping up the last section where we talked about how you have the tapeworm. It has several sections. The head is the scolex. It has that crown of thorns around the top, the suckers that suck up nutrients, and then how it had those individual body sections. Does anybody remember what those body sections were called? Proglottids, yep. And each one of those proglottids were special because they were infectious. They have both the male and female reproductive organs, and they just crank out a bunch of eggs. And so we talked about how during an infection, you're going to release a bunch of eggs, right? If we're talking about cattle or pigs, for example, those eggs are going to be released in the feces. Any other animal that comes along, like their cow friend, and eats some grass next to that contaminated grass is going to get the infection. The larval stages of the tapeworm go and bore themselves into the muscle. And that's where we have to be concerned, right? Is that the larval stages that encyst themselves into the muscle, that's infectious meat. And so that's why I want you to understand a little bit about the tapeworm life cycle, because if I just said that the tapeworm spends its adult life in the small intestines of a cow, you might be sitting there wondering, how do I get a tapeworm infection then? And again, I use the examples of cows and pigs a lot, but there are several other animals that can get tapeworm infections as well, and we'll cover those later in the semester. So I just wanted you to remember that and have a basic understanding. Do you have any specific questions on the tapeworm, the infectious cycle, any details like that?
So from you go ahead. I was just saying, so it's in the muscle of the animal, right? Like it doesn't get into our muscle at all.
It would if you were to get a tapeworm infection. You could get encysting into your muscle as well. But no, typically, if you were to ingest an egg or even that larval encysted stage, it's going to more or less hatch in the GI tract. And at that point, it's going to form that adult worm inside of you, and that's how people get tapeworm infections. They lose weight, or the hallmarks are that people are going to lose a lot of weight. They'll be eating normally, but they're just losing a lot of weight. Normally, they don't have too much digestive distress unless the worm gets really long. These worms can get up to 5 meters in length, and that's when they can maybe in some very rare cases cause bowel obstruction. Normally, before there's any type of bowel obstruction, though, tapeworm, that the person will see tapeworm in their bowel movement, and that's when they're going to go to the doctor, or if they're just losing a bunch of weight. So, good questions. There are no dumb. Seriously, there are no bad questions in here. That's why we're here.
I'm just looking at the cycle and the picture as a whole. So
Yeah.
when it's in the human, how does it get back to the environment then?
There's really not a whole lot in terms of human to environmental transmission. We are more or less considered accidental hosts. We're not a reservoir host for this. It's mostly going to be your cattle or dogs. Frequently, will get tapeworm infections. You might give your dog's worming or cat's worming medication. But yeah, it's the transmission from us back to the environment really is not that a main route of infection for TNA pisiformis. Something I didn't mention before is that there are cases of vegetarians getting tapeworm. Does anybody remember how? Yeah, very good. Yeah, very good. Fertilizer being used and then not washing your salads appropriately. Outstanding. Again, if you're online, please feel free to ask questions into the question bar while we're doing the live presentation, and I can answer those. Anybody else? Other questions? Yes.
Okay, for the virus, the general characteristics of viruses.
Yes.
It says that the viruses are chemical particles, and I'm not sure if like these 4 things are the types of viruses. Because would non-living viruses be the same thing as attenuated virus?
Okay, it's a good question. And so the question was, is a non-living virus the same as an attenuated virus? And the answer is it is not. An attenuated virus is a virus that we've taken in the laboratory and we've grown it several times in cell lines and stuff like that. When viruses are grown in cell lines for several passages, so maybe dozens to hundreds of times, they actually become less infectious. They cause less illness. So even though the virus is still active, it is still able to cause infection, it is still able to replicate, it is no longer causing the same amount of the robustness of that disease. The analogy I use of an attenuated virus would be a dog that has a muzzle on, right? A dog can bark, it can growl, but it can't bite you if it has a muzzle on. All right, that's the same thing as attenuated virus. Attenuated virus is still able to replicate, it's still able to make copies of itself, do the things that viruses do, but you're not going to get the full-blown illness. So if you hear of somebody getting an attenuated vaccine, for example, people use the mistake and say that it's a live vaccine. It's not. It's an attenuated vaccine. If you're getting an attenuated vaccine, you might experience some illness, right? You might get a little achy, you might get a little sore, because you are getting viral replication, just not to the same robustness as you would. This is also why you have to be careful. For example, when you go to the pediatrics office, moms always want their kids to get the nasal spray vaccine when they get the flu shot. You have the shot and then you have the nasal spray. Moms always want their kids to get the nasal spray because they're not getting a shot then, right? Well, the difference between the nasal spray and the shot itself is the dosage, and one of them's active and one of them's not active. So you have to be really careful and ask questions like, do you have other kids in the house, right? Do you have somebody that's elderly or immunocompromised? Because even though you give somebody an attenuated live vaccine strain, there could be some other susceptible person in the house. So asking the patient questions is really important. That's all kind of extra information, but yeah. So viruses aren't live or attenuated, right? They're not live ever. They're attenuated or inactive. And so I think what you're trying to get at is I asked, like, what are the properties of a virus?
Yeah, and this was non-living, but then at the bottom it's active or inactive.
Yeah.
Alive. So,
Those are the words that you use. Instead of saying living or dead, or alive or dead, you say active or inactive.
but then there's one on here that is non-living.
What's that?
There's one on here that says non-living.
Well, yeah, because they're not. They don't meet the definition of life. Viruses do not meet the definition of life. So you'll hear these debates happen a lot on the internet. Well, people will say viruses are live or viruses are dead, and it's like that they don't meet that criteria. Yes.
Do you actually like them? How much of the history of the viruses do you have?
I'm not super concerned that like exact like names of crazy Russian guys, right? That's not as important to me. I want you to have a general idea of like just a little bit on the history in terms of like how did we get where we are today? A lot of people think that we've known about viruses for a very long time, and we just simply have not. We have not learned. Basically everything we know about viruses for the most part has been since World War II. And so having that understanding of, okay, things started off in tobacco mosaic virus, right? We had these living contagious fluids that we called poisons. We didn't really understand how they worked. Like that's more important than me saying who originally discovered X, Y, or Z. That doesn't matter in terms of your health science career. Again, if you have any questions and you're watching the live stream right now, please go ahead and ask and I will get to them. What else do we have?
Can you talk about the differentiation between enzymatic factors?
Yeah, okay. So the point of that question is just to say, do you know what enzymes are? What are the characteristics of enzymes? And so if I say, what is an enzyme? What is it? Well, it's a catalyst. That's a definition. They're often used as catalysts. Okay, that's fair. If you say that it's a catalyst, what does that mean? Yeah, it's speeding up reactions. The typical one that I use in class was the DNA example, right? DNA has a half-life of 10,000 years. It's not going to unfold on its own unless you have an enzyme to unfold it. So it's speeding up the reaction. That's good. What else can you say about enzymes? They don't become part of the products, right? And so the example I used in class was if I was an enzyme and my job as an enzyme was to break down a marker into that, I didn't become part of those products, right? I did not become the product. I interacted at an active site, which I use as my hands, with a substrate. So the whole marker is considered the substrate, the thing that the enzyme interacts with. But when you break it down, you're breaking it down into the products. Good. I didn't become part of the product. What does that mean? Yeah, I can be used over and over and over again. Okay. Can you use drugs to block me from working? All right, what are those called? Inhibitors. And where do those inhibitors oftentimes bind? Yep, to the active site. So an example I could use of that, let's say I take an inhibitor and the inhibitor does that. All right, well, I can't interact properly with my substrate anymore. So that's competitive inhibition. It's blocking it. Okay, good. What else do we know about enzymes? Do I need anything else attached to me? What is just the core enzyme called? Yeah, the apo enzyme. Okay. Do I have things that attach to me? What are those called? Cofactors. Okay, or coenzymes. Very good. So I just want you to know what those are. Like, why do they matter? The example I gave you was essential vitamins and stuff like that in class. So as long as you have that basic understanding, because we'll be talking about receptors, we'll be talking about proteins, we'll be talking about enzymes for the rest of the semester. And I just want you to have that very basic understanding of what that is so I can build off of it. Yes? Typically, yes. Viruses are typically smaller than bacteria. There are a few exceptions, like the example that we gave in class was Mimi virus. Mimi virus was again discovered in 2014. It still lives inside of cells, just like all viruses do, but it is bigger than Rickettsia, which is a version of a bacteria. But yeah, typically the vast majority of viruses are significantly smaller than a bacteria. Great question. Yes.
Can you reverse back to what is a substrate? I didn't understand.
Okay, so the question is, what is a substrate? And that's a great question. So the definition of a substrate is the object in which an enzyme interacts with or acts on. Okay, so proteins, enzymes, have very, very specific substrates that they interact with. So for example, there might be in a solution several different substrates, and I'll just use these as examples. All right, let's say all of these are substrates. As an enzyme, my specific enzyme, right, my only job is to take caps off of markers. That's it. That's all I do. Okay? I do not interact with erasers. I don't interact with rulers. I don't interact with glasses. I don't do anything else other than take caps off of markers. Those markers are then considered the substrate for this protein, for this enzyme. Does that make sense? Okay. So again, that substrate enzyme interaction is highly specific. What else we got? Yeah.
On this little chart thing, it says that matrix protein enzymes are not found in all viruses.
Yeah.
What are matrix?
Very good. What are matrix protein enzymes? So this came out of the virology lecture. There was a slide that said covering and core, right? Covering and core. We talked about how some viruses are DNA viruses and some viruses are RNA viruses, but never both. And we talked about how sometimes those viruses need to bring in proteins to help them out, enzymes to help them out. The examples I gave you on your slides were DNA and RNA polymerases. I think I talked about RNA-dependent RNA polymerases, which are also called replicases. We discussed reverse transcriptases in the case of HIV, where HIV can take an mRNA, HIV is an mRNA virus, and reverse transcribe that back to DNA. Those enzymes, those three enzymes, the polymerases, the replicases, and the reverse transcriptases, those are all three examples of matrix proteins that viruses can bring in. So let me go ahead and draw that out for you. So if I have a virus, let's draw a naked virus. This naked virus has a capsid. Okay. That naked virus is also going to have a genome, shown here in green. You might have on the surface spikes, also called glycoproteins. Remember, those are the keys when we talked about the lock and key mechanism. And in addition to that, some viruses will have these little enzymes in there inside the capsid that come along with the virus. So again, they're shown here in black or blue. These are small proteins. Again, they can be polymerases, replicases, or reverse transcriptases. But again, they come in with the virus. We didn't really go into the details of why that's super important, but it plays a big role on whether you're dealing with a DNA or an RNA virus. There's polarity to these that you don't have to know the details of. I just want you to know that sometimes they bring a little something in with them.
What did you call replicases? You said they were.
Replicases are literally mRNA copy machines. mRNA copy machines. And let me show you why that's important. Normally in a cell, you go from DNA to mRNA to protein, right? That's what you do. This is what cells do. Cells, normal human cells, or eukaryotic cells of any kind, have no way of copying mRNA. Okay, so if you have DNA, you go from mRNA to protein. It's just literally that's forward. The problem here is if you're an mRNA virus, right? If you're an mRNA virus and you get into a cell, your only job as a virus pretty much is to do what? If I said you're a virus, you have one job. What's your job? Infect and make copies of yourself, right? I just want to be a copy machine. Well, these mRNA viruses have a problem. They're made of mRNA, and your cells have no way of making copies of their mRNA. And so sometimes what they do is they bring in mRNA polymerases or replicases is another term that you can use for them. And what those replicases, let's say that this replicase here, let's say this is an RNA replicase, right? What's going to happen is that this is inside the cell. So let's imagine that we're inside. Here's a cell, here's a nucleus. We are in the cytoplasm here of a cell. This mRNA replicase, the virus has gotten in. We've done adsorption, right? We've done penetration, entry, uncoding, all of that. Now we have to do that amazing step of synthesis, right? And part of synthesis is replicating. Well, this mRNA synthesis, this mRNA replicase is going to read this mRNA and make a copy mRNA. Copy, 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 copy. And so, again, a normal cell can't do that, so the virus has to bring in a protein to make copies of the genome. Does that help make sense? Good.
So is the only difference between a replicase and a polymerase the fact that polymerase does both DNA and RNA?
Yeah.
And replicase is just RNA?
Yeah.
Okay.
Yeah. Good question.
Can you explain the role of a phage in adsorption? Or what it is?
Okay, so again, a phage is referring to a bacteriophage. So we talked about how some viruses infect bacteria. So let's imagine that this is a bacillus here. If you remember your phage viruses, they look different. They look kind of like spiders. So they might look something like this, right? That spider-looking lunar lander object does not get absorbed into the bacteria. All right. If you remember when we talked about it, what's going to happen is this spider is going to crouch down. It's going to poke a hole inside of the cell wall of the bacteria, and it's going to take this genome and it's going to inject the bacteria with the genome. So phage viruses are not absorbed. Phage viruses are not absorbed. Again, your normal virus, if we talked about a traditional virus, like if we had this naked virus over here and we had our cell over here, this naked virus has to first attach to a receptor on the surface. That's the lock and key mechanism. And then it has to be absorbed. All right. And if it's a naked virus, you remember that that's going to go through endocytosis. And so at the end of the day, you're going to have a virion that is endocytosed. And again, it'll look something like this. So a normal naked virus goes through attachment, right? That attachment and absorption phase, whereas a naked or a phage virus has no absorption whatsoever. It's just landing on the surface of the bacteria and physically injecting its genome. You see that difference? Okay, good. Other questions? Yeah, that's good. Yeah, that's a good question. All right, so what I didn't really have time to explain this in class. So let me zoom in on this part of the phage. So if I were to zoom in to that part of the phage, you have the sheath, which I described in class as an accordion. And then off the bottom of that sheath, you literally have teeth, all right, that will puncture into, when this virus squats down, it will puncture into the cell wall. So here's your cell wall of your bacteria right here. So what's happened here is the phage squats down and these teeth puncture the cell wall. This is kind of like if you were to shove a shovel into dirt, right? Did you just break a giant 50-foot hole into the ground? No, you just literally shove the shovel into the dirt. Well, now what this phage does is it has lysosome enzymes that are up here in this accordion piece that basically, think of it as kind of like snot or drool. It literally kind of coats and leaks around these teeth. And it's just an enzyme. And what this enzyme is doing is dissolving away the peptidoglycan. It's dissolving away the phospholipid bilayers. And so when those lysozymes are done doing their job, what happens is that there is literally a hole in the cell wall that has dissolved away. So it's literally dissolved everything away, and it creates a tube, all right? It's like a hypodermic needle. And what it does is that's what allows the genome to be injected when the sheath contracts, is because you're injecting the genome through a tube. So your mRNA or DNA, whatever, comes through the center of that tube into the cell. But it's the lysozyme that's physically dissolving away that cell wall. Great question. Yes?
And that's for
Phage
bacteria
viruses.
that has an envelope around it, or
No. So this is not for bacteria. This is you're talking about gram positive versus gram negative.
Is it just like, why would a phage be used instead of like?
Okay, so phages are a completely separate category of viruses that only infect bacteria.
Okay, it doesn't matter what kind of bacteria.
It does. Different bacteria have different phages,
Yeah.
but they attach using these these feet, they're called tails. That interaction is specific. Remember we talked about glycoproteins over here, or spikes. The tails are specific to each bacteria on those phages. But phages only infect bacteria. They don't infect human cells. It's actually kind of neat. Russia, back in the Cold War, when we were really developing antibiotics, so post-World War II, pre-Cold War, the United States said, oh, antibiotics are amazing. We discovered penicillin. Let's start making antibiotics. And Russia said, oh, that's okay, but we're going to actually develop phages. And so they actually developed phages just like we developed antibiotics to try to fight infections, because they said, hey, there was no way that the human cells are going to be killed by these phages. They only infect bacteria. So it was a great idea, right? I could give you a shot of phage and kill whatever bacteria is inside of you. The problem that they ran into is several decades into their studies, they had to give up because they found that bacteria become resistant to phages faster than they do antibiotics. And so that's why you don't hear about it in the news. You don't hear about the new phage antibiotic that's come out, because resistance is too fast. Again, for those of you viewing on YouTube, please type your questions in the comment section, and I will answer them. Yes, question.
So is host range basically the same thing as like the lock and key, like you have to, it has to go together?
Yeah, so the question was, does host range have to do with anything? Oh, this board already has stuff on it. Does host range have to do with anything with the lock and key mechanism? And it does. It's pretty much the exact same concept. So in class, we talked about broad host range, intermediate host range, or a pretty narrow or restricted host range. And that was the lock and key mechanism. Again, each virus has its own glycoprotein on the surface, and the glycoprotein on its surface will only bind with a specific receptor. And that lock and key mechanism, again, it has to be 100% specific. So if you remember the difference, what was the difference between a broad host range then and a narrow host range? Why can they affect more? Because different cells sometimes share the same receptors. And so if I were to draw that out, let's say I had a liver cell, a kidney cell, and a nerve cell. Okay? The liver cell might express blue receptors. And every cell in your body expresses different receptors. It's part of what identifies that cell as a specific type of cell. So maybe liver cells have blue receptors, kidney cells have red receptors, and nerve cells have green receptors, right? That makes sense. Well, if I come over here and I make a virus, let's make a virus, all right? And I put glycoproteins on the surface of this virus, and let's say the virus only expresses those glycoproteins, what cells can I infect? Liver. Those are the only cells that this virus can infect. Now, check this out. Sometimes cells, so viruses will have a glycoprotein on them, but cells can express sometimes hundreds of receptors on them. So let's say, in addition to those receptors, let's say liver cells, nerve cells also express this kind of light green receptor on them. If you crank out a virus, okay, what kind of cell can that virus infect? Liver and nerve. Very good. So you see, it's just what receptor does the glycoprotein bind to? So this is the glycoprotein. Those are the host cell receptors. And so depending on what glycoprotein that virus has on its surface completely dictates what cells can be infected. And that's a really neat concept when you think of virology, right? Because people are always worried about contracting viruses. You know that this specific virus only can infect GI cells in your GI tract. And somebody sneezes on your arm. Are you going to get that virus? Probably not, unless you rub your arm and suck on your fingers, or rub your arm and eat a sandwich that has the virus on it. You're probably not going to get that infection because of that absolute specificity. So everyone's freaking out about coronavirus, right? Coronavirus, how is that spread? Respiratory droplets like flu. So you have to worry about people sneezing on you. You have to worry about somebody coughing in your face, because any mucus that is leaving their body potentially will have virion particles in it. All right, great. Any other questions? Yes.
For the glycoproteins.
Yes, the glycoproteins.
So they can be found on both a naked virus and an enveloped virus.
Very good.
Are they always on an enveloped virus though?
Yes. Here's the difference. Okay, so here's a naked virus. Let's draw an enveloped virus. The enveloped virus is still going to have a capsid, just like your normal virus does. However, it's going to have that phospholipid bilayer on the outside. The reason it has that phospholipid bilayer on the outside is because it's budding. This is a budding virus. This is a lytic virus. And so this one, the glycoproteins will not be on the capsid because it's inside of an envelope. They'll be out here on that phospholipid envelope bilayer. So what type of cell does that virus infect? Kidney. Very good. But do you see that difference?
Yeah. So, but a naked virus does it still have to have glycoproteins on it?
Yeah. So because if you had a virus, so the question was, does a virus have to have glycoproteins on it? So if you took a virus and you did that to it, where it had the capsid, like it could have the capsid, it could have the DNA, it could have everything on it. If it doesn't have glycoproteins, it cannot bind to a cell. Okay, and this is actually a pretty important piece, because many vaccines that are produced are actually targeting those glycoproteins. Because if I were to inject you with just those glycoproteins, your body could make antibodies against those glycoproteins. So then what would happen is those antibodies would come in and they bind to the glycoproteins, coating all of the glycoproteins with antibody. Do you think that virus can bind to a host cell? No, because all of the glycoproteins are now coated with antibody. This is why many of your vaccines work the way they do. Great questions. Yes.
So for like when the virus is released, like after it's replicated and
Yes.
everything. So for the lysis, do naked viruses always do lysis?
Yes. Yeah, naked viruses always do lysis, because what's happening here, and I'll bring our cell back down. What our naked viruses are going to do is they're just going to replicate like crazy. And so this is obviously not to scale. But what happens is these viruses just make copies and copies and copies and copies and copies of themselves until literally the cells burst. And they burst open, releasing the naked virus. Now, budding viruses, they don't do that, because the disadvantage to being a lytic virus is that you're causing tissue damage. You're going to cause damage to the host, you're going to get inflammation, you're going to have to fight the immune system a little bit more aggressively. That's not so much fun. So what other viruses will do is they'll say, okay, I am going to make my capsid, but then I'm going to bud off of the surface. So in the process of budding, you get that phospholipid bilayer, which is literally host cell membrane wrapping around that viral particle until it pinches off. Now, that whole process is very complex. We don't need to know the details of it. But I do want you to know that this outside coating, that envelope, is literally just host cell membrane. It's host cell membrane. The advantage to doing this process is the cells don't explode. You could maintain an infection for a really long time and just be a budding virus. That's a nice piece, right? Fun fact, you don't have to know this, but it's kind of a fun fact. You know, when you take Tamiflu, right? Tamiflu, if you give it within 48 hours of getting the flu virus, influenza virus, it will make your symptoms go down. The reason it makes your symptoms go down is what Tamiflu does is that there is a process, there's actually proteins involved in pinching off of that membrane. When you take Tamiflu, what Tamiflu does is it stops budding from happening. It stops that pinching off. And so if you were to look at a cell of a Tamiflu infected person, they would have a cell that's infected with virus. It is replicating, but it's not pinching off from the surface. So this would be kind of like if you had a pack of bulldogs and you had them on leashes. They're attached. They can't go attack anyone. They can't because they're stuck. They're chemically stuck to the surface of the cell. Now, what happens if you're lazy and you don't take your Tamiflu? You let go of the leashes. All these viruses start budding off, and you get infection. So again, that's extra information. You don't have to know that, but it's fun to think about. Yes.
So envelope viruses don't by budding.
Yes, envelope viruses only leave a cell via budding. Very good.
A virus can have an envelope covering or a capsid covering? Or is it
Well, they will always have a capsid. So the question was, do viruses then only have an envelope or a capsid? And the answer is no. They have either they all have capsids, but some of them have envelopes. So all your viruses are going to have capsids, but only some of them will have envelopes. Make sense? These are great questions. Again, if you're online, you have any questions for me, please type it into the comments section and they will pop up. I don't have any right now in the live chat. Question?
Can you explain a little more on the virulence factors?
Virulence factors,
Yeah,
yes.
those.
Yeah, no problem. Okay, so let's talk about an infectious bacteria. Let's talk about Clostridium botulinum. All right, Clostridium botulinum is the bacteria that causes botulism. If you've heard of botulism before. So botulism, let's talk about just real quick how botulism works. So if you remember from AMP, you have a neuron that interacts with a muscle at the neuromuscular junction. Does that sound familiar? So you have a neuron up here, neuron, and muscle down here. Normally what happens in that neuromuscular junction is the nerve gets stimulated. It releases acetylcholine into the synaptic cleft. That acetylcholine binds to an acetylcholine receptor, which causes the muscle to depolarize, if you don't remember from AMP. What happens if you get botulism, what's happening there is that you have bacteria somewhere in your body, and those bacteria are releasing botulism toxin, which I'll represent here as little green dots. What happens is botulism toxin builds up in the synaptic cleft and around this terminal end bulb, and it prevents the nerve from releasing acetylcholine. Okay? So the toxin is preventing the nerve from releasing acetylcholine. Does that make sense? So people that have botulism they are not going to be able to contract their muscles. Okay, in adults, this can cause things like blurred vision. It can cause, in severe cases, respiratory distress because you can't contract your diaphragm. In babies, it causes a condition called floppy baby syndrome. Because the babies can't contract their muscles, they lose what's called their flight response. You know what a flight response is? It's where you do this when someone picks you up or whatever. Babies won't do that. We'll talk about that more later. Okay, now you get that concept. So this toxin is important for this bacteria when it makes you sick. Well, if we were to zoom in on the Clostridium botulinum, we were to zoom in on this bacteria, it has several genes. So we'll just look at the DNA inside of this bacteria. Some of the genes in the bacteria are always going to be on. They'll always be produced. Okay? These are genes that are important for things like making of ATP, so glycolysis, right? Making of peptidoglycan, PGN. Those genes are always on, so they're called constitutive genes. Always on. It's always important for the cell to make ATP, right? Now, some of the genes, like the toxin gene, are only turned on under special conditions, like when it's infecting your body, right? Those genes that are only on under special conditions are called regulated genes or virulence factors. A virulence factor is anything that allows a bacteria to survive in the host better. In this case, it's Botox, right? Botulinum toxin. In other bacteria, they could be other things. Like if your bacteria produces a capsule is considered a virulence factor because the capsule is anti-phagocytic. Does that help clarify that a little bit?
So are regulated genes and regulated enzymes the same thing then?
Regulated genes, a gene, so this comes back to our central dogma of biology. You have DNA that turns into mRNA that turns into protein, right? Genes are at the DNA level. Gene. Enzymes are proteins, all right? So what happens is if you activate a regulated gene, you're going to make the mRNA for that gene, which will cause the protein to be made for that gene. So, do you see that difference?
Yeah, that makes sense.
Very good. Fantastic questions. These are great. Yes?
What were the two examples that you gave in class about that? One was Lyme disease and I couldn't hear.
So the examples I gave in class of bacteria having regulated genes were I used Lyme disease, and let me think of another one.
The one that you just used.
I used Clostridium botulinum as well. I mean, there are hundreds or thousands of examples that I could use there. I wouldn't get hung up on those examples specifically, but more so the concept of a virulence factor is allowing the bacteria to survive better in the host. Great. Again, anybody viewing online, you're more than welcome to ask questions in the comment section. I don't have any yet, but that's okay. Yes.
So on the last page of the virology packet, it
Yes.
talks about damage to the host cell and persistent infections, and there's three things listed: the cytopathic event, inclusion bodies, and
Yes. Yeah.
syncytia.
Syncytia, yeah.
Syncytia. Are those all essentially the same thing, or different ways of reading it? If they sound similar?
They're very similar. What I want you to know about those is that's how you would clinically diagnose people of having viral infections. It's one of the ways that you can do it. So if I were to look at a tissue sample and I were to see syncytia, which is multinucleated giant cell formation, that tells me that there's a viral infection going on. if I'm in a lab and I'm maintaining cell lines and I see inclusion bodies in the cells, that tells me that there's cell replication going on. These are just different pathologies that cells express. And the reason that I even talk about them is that they get written down on charts. So if you're reading a chart, it might say syncytia, or it might say multinucleated giant cell, or it might say inclusion bodies were observed. I just want you, when you're working in the lab setting, to be able to see, when you see something like that, you go, oh, boom, I know that they have a viral infection. It's just a trigger word to try to get your brain to go click. I know they have a viral infection. I don't necessarily know what viral infection. Does that make sense?
Yeah.
They're different pathologies from viral infections.
Okay. Yeah.
These are great questions. Is there anything that you're worried about with tomorrow's exam other than, or the next day, depending on what class you're in? I know these are coming quick, but then we have less material that we're covering in each exam, so it's kind of nice from that aspect. I'm just checking to see. Okay. No questions are showing up there. Yes.
Do you send out like an email about the link, or like how do you know like where the link is at? Because I know the one you sent last week, like it was.
Yeah, so if you go onto Canvas, like you could do this right now, if you go onto Canvas and you go to the announcements that I sent out yesterday under the announcements section, there's one that's going to say section 3 review session, or exam 2 review session, I think is what it says. The last part of that, there's a link that says click here. If you click there, it will pull up this presentation that's currently happening. And so let me know if you're having any problems with that. What's that? It worked for you? Okay. I pulled it up on my phone before I started talking. So, and then once I get done and I kill the live stream here in a minute, it'll just be a video that you can play back. So I'm sure that you could continue commenting, but I'm not going to be talking anymore. So
I just got the introduction to microbiology exam 2 study guide.
Yeah, I
Should
never
there be
got
a
the-
message from
yeah. So if you go to the Canvas page, because I can do it right here. If you go to the Canvas page, under announcements, it says the latest announcement that I sent out says exam 2 study session. In that exam 2 study session, it says what, when, where, and then it says live stream. At the bottom where it says live stream, you click here and it takes you to me talking.
I don't. Yeah, I still don't have that like announcement.
The recent one is the one about the study guide. That's the only one that
Yeah.
I have. Me too. So maybe we're not in class.
Yeah, because I don't know, that's weird.
I did it.
Because I checked, because I was like, gonna just do live stream.
I only knew from last time. So.
I would log into.
Well,
So you have something weird
we're
set up
all
with
on
Canvas then.
Wednesday, Friday. Do you have different pages for Wednesday, Friday, and?
Yeah, but I have it in both sections.
I was saying mine shows up.
Yeah. So let me go back to.
Maybe because I can't see like at all.
Are you here?
Like the announcement?
All right, I'll go back and I'll check and see if it posted your class. If not, I'll just post it and you'll have the link.
Okay. Like I don't know what he's talking about, where that is.
Okay. Well, I'll check that here in a minute as soon as I kill this. I'll go back to my office and check it. If it didn't post for some reason, because I did them like back to back, like copy paste, copy paste. All right. Any other last second questions? Does this help? Is this helpful? All right. Well, thank you all so much for coming. I appreciate it.